Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Professor Dr. Arno Villringer. Arno is the director of the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, Germany, director of Department of Cognitive Neurology at Leipzig University Hospital, and academic director of the Berlin School of Mind and Brain and Mind and Brain Institute in Berlin. He holds a full professorship at the University of Leipzig, an honorary professorship at Charité, Humboldt University of Berlin, and an adjunct professorship at the Medical University of Vienna. Dr. Villringer received his medical doctorate degree in molecular biology from the University of Freiburg and subsequently completed a fellowship in NMR unit at the MGH, Harvard Medical School, supervised by Tom Brady and Bruce Rosen. He then moved to Berlin, working at Charité University Hospital, and received an endowed professorship at Humboldt University. Clinically, he served first as senior physician and later as the head of Department of Neurology at the Benjamin Franklin campus. In 2006, he founded the Berlin School of Mind and Brain and has since been its academic director together with Michael Pauen. Since 2007, he has been the director of Department of Neurology at the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig and director of Department of Neurology at Leipzig University Hospital. While in Boston, Dr. Villringer pioneered magnetic resonance perfusion imaging of the brain, showing that susceptibility contrast agents such as GDDTPA could be used in MRI, which later also became relevant to fMRI. In Berlin, he later developed non-invasive perfusion imaging based on a bolt delay approach. Dr. Villringer is also known for pioneering and validating functional near-infrared spectroscopy and imaging of the human brain. His current research focuses on brain-body interactions. He is pursuing the overarching hypothesis that dysfunctional brain-body interactions are critical to the development of obesity and hypertension, which eventually lead to stroke and dementia. The somatosensor system serves as a model system for his group to study brain-body relationships and to understand basic biological mechanisms underlying brain-body interactions. Dr. Villringer uses behavioral approaches in combination with multimodal brain imaging techniques, including fMRI, EEG, MEG, FNIRS, and TMS. I would like to remind the audience to please address any questions using Q&A box or raise their hands at the end of the talk. If you have anything you want to clarify, you can also just let us know during the talk. Dr. Villringer, thank you very much for coming, and the virtual stage is yours.
Thank you very much, Ishil, for the very kind invitation and the very kind introduction. It's a great pleasure for me to be with you. And so I will start now. And actually, this morning, when I wanted to take a train to Leipzig, I just wanted to leave the house, and then I realized I didn't find my glasses. I was walking around hectically, I just didn't find them. And just by accident, I looked into a mirror, and lo and behold, I saw the glasses on my nose. The same skin receptors that had not told me about the glasses on my nose now actually let me feel the glasses. I'm sure many of you have had similar experiences, and if not, I'm sure you will have them sometime in your life. In any way, welcome to the gray zone of perception. This is the kind of things that I would like to talk today. Now, when we talk about perception, we typically have a quite simplified view. We have a sensory organ which enters the signals enter the body at first, or the brain. At first it's unconscious, and then somehow it comes to conscious perception. And I want to talk about something in between, something that is not easily dichotomized into conscious versus unconscious. And that's what I call the gray zone. And I will talk much more about what I mean with the gray zone and different ways how to define it. Also, you see the term conscious, unconscious here. I will try to avoid the term consciousness as much as possible, rather replacing what I'm talking about by operational definitions, what I mean. So, the gray zone of perception. In my talk, I will discuss cognition, behavioral aspects. I will discuss neural correlates. I will also discuss neural correlates of entering and exiting the gray zone. And finally, I will also discuss how the body can influence processing of stimuli in the gray zone. So the sensory modality I am interested in is the somatosensory system. And as you can see here, some of the stimuli are not easy to operationalize, but therefore we are using a quite simple method of stimulation, which is an electrical finger stimulation. It's very localized, obviously, and it's also very well defined in time, a perfect method to investigate processing in space and time. We use behavioral studies, including signal detection theory. And when I say we look at neural correlates, we mainly do this with EEG and fMRI, as you will see throughout my talk. Also, just to clarify nomenclature and operational definitions, what you see here is a very general psychophysical sensory detection curve, very schematic. So if a stimulus is very, very strong and that we always feel it, then we call it suprathreshold. If we never ever feel it, it is below the absolute threshold, I call it subthreshold. And then there is an interesting domain of stimuli that we sometimes feel and sometimes not. And there is this sigmoid detection curve, which is quite typical for many sensory modalities, which I call near threshold stimuli. And since those latter stimuli allow us to apply the same physical stimulus, but then sometimes have it perceived or not, it's quite interesting, and it's a typical question that we asked, are there different neural activation patterns of perceived versus unperceived near threshold stimuli? I'm going to report some of the recent experiments that we did on this. First, let me look at fMRI, actually studies done by Martin Grund recently, and he applied near threshold stimuli and did fMRI and then compared undetected versus detected. More precisely, in this comparison, he looked at those stimuli that were not detected, undetected stimuli, but they were present, in contrast to stimuli in contrast to catch trials, which were correctly rejected. And what you see is that when you have this contrast, so there's a stimulation not detected, a near threshold stimulation, then you see S1, actually that's not seen here, but it's present. You see bilateral S2, you see SMA, and you see a weak activation of insula. If you have a stimulus, the same kind of stimulus, the same strength that is detected, and then you compare the detected versus the undetected stimulus, then you have additional activation in bilateral insula, in the IFG, in the precuneus, also nucleus accumbens. If you go a bit further and you use only confident hits, especially the precuneus is much more dominant, and also the PCC becomes very clearly active. So to summarize those findings, which confirm actually also findings in other modalities, if you have a sensory near threshold stimulation, which is undetected, you still have activation of S1, S2, SMA, and a little bit of insula. And if it becomes detected, then you have also these areas, but in addition, so stronger left anterior insula and right anterior insula, and then a frontoparietal network, IPS on both sides, precuneus, IFG, and nucleus accumbens. Now, this is confirming and also showing for the somatosensory system this contrast. I told you before that there's also these subthreshold stimuli. So we asked ourselves some time ago, what about these subthreshold stimuli? How do they fit into this picture? After all, psychophysically, you cannot really differentiate them. They are in a way undetected, like the undetected near threshold stimuli. So, and we also did electrostimulation, and we did fMRI, and when I say we, that was Felix Blankenburg who started this experiment. First, we wanted to confirm that subthreshold really is subthreshold, so that they are not more often detected than a catch trial. And that's what we showed here. So this is the number of false alarms, and you can see that both these are two different types of subthreshold stimulation, but the one that we used and the other one did not exceed the false alarm rate. So they're really subthreshold. And interestingly, when Felix did this experiment, he did not find any activation in the brain, but rather he found in S1, contralateral S1, S2, and SMA, he found a deactivation. You see in this slide again, summarized, which was very surprising to us. And the question is, what does it mean if there is a deactivation? Just to put it in the picture, I showed you before the near threshold trials, and basically the same areas that are activated in undetected near threshold are now deactivated in undetected subthreshold stimuli. What does it mean? So in order to further our understanding, we did also EEG studies on the very same stimuli. So we again used near threshold stimuli and subthreshold stimuli. And first we compared detected versus undetected near threshold, and then we looked at subthreshold stimuli. So when again electrical finger stimulation. This is a typical evoked response to a near threshold stimulus, work done by Norman Forshak. And so there's an N20, I will focus on that later, but especially I will focus on P50 and N150, typical components of an evoked potential, and controversially discussed what they actually mean with respect to detecting a stimulus. And I think there's now a clear answer by Norman, and also it has been confirmed by the group of Felix Blankenburg, basically showing that the P50 does not indicate whether you detect it or not. Rather, it reflects the strength of the stimulus. So these are the strong stimulus, a weak stimulus, and you see it's increasing with a strong stimulus. But there's no big difference, no significant difference between detected, undetected, detected, undetected. However, the N150 was in our hands the first component, and also in Felix Blankenburg's lab by Pierre Schröder, the first component who clearly reflected the detection of the stimulus and not so much the intensity. Now, so P50 scales with stimulus intensity, N150 scales with detection. What about now, again, to this picture, what about these undetected stimuli that have a completely different pattern, a deactivation in fMRI? What do they do in the ERP? Is there any ERP at all? Does it reach? That was a question again. We asked with EEG, we answered with EEG, and the person who did it was Till Nierhaus. And here you see in his experimental series, again, the ERP response to a near threshold stimulus. You see a P50 and 170. So yes, this stimulus was detected. And now what happens to a subthreshold stimulus? What you see here is a similar response as the P50, slightly later, P60, but nothing going beyond. So it seems that these stimuli seem to reach the primary somatosensory cortex, but somehow they stop at this moment. Why do they stop? So this summarizes basically all these findings. So there's nothing beyond the P60 in those undetected stimuli. Why do they stop? Let me go into some behavioral studies that we did on those subthreshold stimuli. This was Gabriel and with Felix and Birol. What we studied now is what behavioral consequences does or are there any behavioral consequences of a subthreshold stimulation? And so we applied subthreshold stimuli that you see here, and simultaneously we applied a stronger near threshold stimuli to detect or to determine the sensory threshold. And so we did it with subliminal stimuli and without. And what we found was that the detection threshold with the subthreshold stimuli was moved to the right. So there was a higher detection threshold, actually for all subjects that we investigated. In other words, what we find is that the cortex was inhibited by these stimuli. Not only does it not proceed, but it somehow seems to inhibit the cortex. Actually, we were able to show that this not only refers to the same finger, but also to the adjacent finger, worked by Birol Tashkin. So when you stimulate the adjacent finger, digit 3, that's what you see in S1, and a typical fMRI activation. If you now add, so you have more energy added, subliminal stimulation of the adjacent figure, all of a sudden it disappears and detection is also inhibited. So how can this be explained? What are potential mechanisms underlying an inhibitory effect of subthreshold stimulation? So a potential explanation comes from the work of Swedlow over many experiments that he performed in rabbits and rats. And what he noted is, now he has schematic interpretation of his findings, when there was a high stimulation intensity in the somatosensory system, forepaw, or then in the rabbit, then he found that, of course, there was an overall excitation of excitatory neurons and an activation of inhibitory neurons, an overall activation of the somatosensory cortex. However, when he used a very low stimulation intensity, similar to the one that we were using, then he finds a preferential activation only of inhibitory interneurons. And this feed-forward inhibition would basically downregulate the entire cortex. So our hypothesis was that this, or is still, that this is the mechanism how subthreshold stimulation not only is stopped for further processing, but also stops or inhibits the somatosensory cortex for other stimuli. So again, a summary of what I have showed you so far. And now also a summary what this behavioral effect is of subthreshold stimulation. So if you have near threshold stimuli, and let's assume we have the same intensity, some are detected and some are not. And when we add subthreshold stimulation, less of those stimuli are detected. Now, this finding, of course, and I will come back to the mechanisms of subthreshold stimulation, but this finding raises the more general question: why is it actually that some near threshold stimuli, even without subthreshold stimulation, sometimes they are perceived and sometimes not? So, and this is a question that we asked in a series of experiments. So we applied always the same near threshold pulse, and we wanted to know, is there anything in the brain before the stimulus is detected or not detected that kind of predicts detection or not detection? And the earliest experiment we did on that was actually, in this case, not a near threshold stimulus, but this was a masking experiment where a stimulus was applied to a finger on the left side, and a mask was applied to the index finger on the right side. These are the evoked potential when the target was detected and not detected. And when we looked at the EEG before, we found contralateral to the target stimulus. Then when the target was missed, we found an increase of alpha rhythm power. In other words, we hypothesized that alpha rhythm power may be a predictor of subsequent detection of sensory stimuli. And in a recent study, Esra also looked at, now she again looked at near threshold stimuli, and she also finds with higher alpha power, the detection of sensory signal goes down. Now we started to look a bit more closer on psychophysical effects of alpha power. and with signal detection theory. I guess all of you know signal detection theory, but let me just give you some details and discuss also what this means for our case. So signal detection theory basically assumes a noise distribution and a signal to noise distribution, and basically the separation, the distance between the two, d prime, is a marker of the sensitivity. And then there's also kind of an optimum of observing it, of putting our criterion, which would be here in the middle. That would be the ideal observer to separate those two distributions. However, in real life, what we find is that our criterion often is not at the place of the ideal observer, but at some distance. For example, this would be a very strict criterion. Only those stimuli would be accepted as hit, and all the others would not pass our detection threshold. Now what is very important, when we cannot measure based on behavioral study the criterion itself, but what we do measure is C, the distance between the criterion and the ideal observer. This is often confused, and in many studies you say we measured the criterion. No, what is being reported is C, and it's becoming very important for what I'm wanting to say right now. So now let's assume the criterion changes, and actually that is what we find. Actually, in this case, I refer to a study by Luca Yemi, but we have had the same findings, but it's a very nice figure by Luca. What he shows is that with stronger alpha power, yes, the criterion decreases, whereas sensitivity does not decrease. So back to the signal detection theory. So what he finds, actually, now I made the mistake myself, not the criterion, but C decreases with increasing alpha power, meaning a more liberal criterion. So what do we think? What does it mean for this figure? So if criterion decreases, if C decreases, we would expect that basically the perceptual threshold just moves to the left. But this is only one possible explanation for C decreasing. The other possible explanation goes like this. Let's assume the criterion in relationship to the sensed intensity of my decision criteria stays the same. But let's assume that the overall excitability of the cortex changes, so that the entire distribution, noise and signal, just move to the right. Let's say it increases the excitability. Then you have something like this. Now, the criterion actually is unchanged, but C has decreased. In other words, when you find a decrease in C with alpha, with higher alpha, it could be either due to a change in the criterion, the perceptual criterion, or it could be due to a change in excitability. And basically, this is a question that we and others tried to address. Does alpha rhythm change excitability, or does it change the decision threshold? There are several indications now that indeed it could be the excitability. I just show you one of them. This is Ole Jensen's group, where they showed Daria Osapova. They showed that the alpha strength inversely correlated with gamma power. So when alpha is strong, gamma power is weak, and vice versa. This is one indication that it could be related to excitability. I think quite nice work has been done on this by Tilman Stefani recently, and that's why I want to go through, tell you a little bit about this work. So what Tilman was measuring was another parameter of excitability, which is the very first cortical component of the SEP, namely the N20. So therefore, in this case, he used a very high resolution EEG device with very, very good signal intensity. So he could look at single trials, N20s, and you see here the time course. And then he checked how pre-stimulus alpha affects the N20. Now, the N20, you should know, is a very, very stable parameter. So you even have an N20 in coma. So to see any effect on N20, which typically is thought as a stimulus-driven parameter, then you really have to have a very, very good signal to noise. It's also a quite small component. Now, what he actually finds is, and I should also mention the N20 is a pure excitatory input. It's the first excitatory thalamocortical input into S1, and it's purely excitatory. And what now Tillman finds is the following. So with increasing, so in the highest alpha quantile, he shows the highest N20. So assuming what others have suggested, that higher alpha is related to lower excitability. So at first sight, it might be surprising that N20 with lower excitability is higher. Here, but then he used structural equation modeling to assess whether it really does, in fact, relate in this fashion to the initial cortical response. He also used a paradigm with different stimulus intensity, and stimulus intensity as well as alpha power both significantly affected the initial cortical response, which then, in this case, was also reflected in perceived stimulus intensity. Now, why is it lower? Why is the N20, which lower in a presumable state of higher excitability? Now, EEG, as we all know, does not measure action potentials. What it does, it reflects synaptic activity and the transmembrane currents, basically, that you need until the action potential is elicited. So now let's have a look at the resting state. In the resting state with average excitability, membrane potential is -70. And so you have transmembrane current until from -70, the threshold for the action potential is reached. And this is the schematic evoked potential. In a hyperpolarized state, then you are at -90. So the distance to the threshold for action potential is much higher. And so you need a higher transmembrane current to reach that. And if you have high excitability, it's the other way around. So in a state of high excitability, you expect the transmembrane current to be lower and therefore be associated with a smaller evoked potential. So it precisely fits. And so I think these data nicely give an additional evidence for the somatosensory system that alpha rhythm strength is really inversely related to excitability. And so I'm concluding that alpha rhythm is inversely related to cortical excitability and not the decision threshold. And with higher alpha power, less stimuli therefore are detected. And now coming back to having said this for the spontaneous variability that near threshold stimuli are detected, let me come back now to the undetected subthreshold stimuli and still address a question I have not answered before. Do we have any index for this inhibitory effect of those subthreshold stimuli? And I mentioned before that we think that this is due to a feed-forward inhibition. Now, this feed-forward inhibition, one would claim, if this is the case, it should inhibit the somatosensory cortex. And if alpha rhythm is a marker of excitability, then alpha rhythm should go up. And that's what we tested. So does subthreshold stimulation modulate alpha power as an index of cortical inhibition? This is experiments done by Thiel Nierhaus. And what he did, he first performed a suprathreshold stimulation, looked at EEG, in this case rhythms, and so this is evoked potential, and what you find here is what everybody knows. After the evoked potential, in the frequency domain, you have a pronounced desynchronization of alpha. So that's very well known. Alpha is desynchronized, goes down after a suprathreshold and also near threshold stimulation. What happens with subthreshold stimulation? Precisely the opposite. So with subthreshold stimulation, alpha transiently increases. Also statistically significant. So this goes very much in line with the transient inhibition of the somatosensory cortex as we had suggested. And here you see an entire head plot. So you see that suprathreshold stimulation actually leads to a widespread desynchronization and subthreshold to a contralateral synchronization increase of alpha power. Now, is this just something specific for the somatosensory system, or is it present in other modalities like vision? At the time we had these findings, I had an office very close to Nico Busch's office, and he said, I don't believe anything in the somatosensory system. It has to be proven also in the visual system to be relevant. So we joined forces, and Isabel Barreiter, a PhD candidate, basically did the same kind of experiment for the visual system. So to make a long story short, she applied subliminal or subthreshold stimuli for the visual system. She found an inhibition for subsequent stimuli. And also, if she looked at in the frequency domain, she also for the visual system found that with supraliminal stimuli, you found a desynchronization, well known, but with subliminal stimuli, subthreshold stimuli, she found a transient increase in alpha rhythm. So this is good evidence that this mechanism, which we think is a noise suppression mechanism, so the brain interprets these very weak stimuli as noise and then it shuts down in order to let the noise not through. We think that is a generic mechanism. So to summarize what I have said so far, alpha rhythm indexes cortical excitability. It seems to determine the transition to perceptual awareness for near threshold stimuli by modulating this excitability. Subthreshold stimuli actually seem to elicit or to strengthen alpha rhythm, also then to modulate near threshold stimuli. And of course, very strong stimuli almost always go through. Now, is this the entire story? Is this all the story about rhythms or determinants of perceptual awareness? Now, if we look a bit more closely, and I did this some time ago at our earlier studies, we not only saw alpha rhythm, but we also saw indications that also some changes in beta, here at CBZ, the central beta, but also frontal beta, could also be related to stimulus detection. And also in a recent study, Norman again found alpha to be relevant for stimulus detection. I've shown this, but he also found a beta band effect, which also pre-stimulus was relevant or determined stimulus detection. Now the question, of course, arises very general. So I told you that alpha seems to just shift this distribution, but the question arises: is there also maybe some parameter which changes actually really the perceptual criterion in a way that without changing the excitability? And so the question is, can we induce a top-down change in criterion by changing, for example, expectation of stimuli occurrence? So by this, if we can induce this and change, again, signal detection criterion, maybe this affects higher order mechanisms. And these are actually current studies that are ongoing, and Karina Forster is doing them, and she just gave me her first results. So basically what she did, she modulated in this experiment the stimulus expectation. So in one condition, you were expecting many real stimuli as opposed to sham, and in the other stimulation, fewer, only 25% real stimuli as opposed to catch trials. And lo and behold, this really changed the criterion of the subject and not the sensitivity. So when you are expecting many stimuli, when you have then subjects lowered significantly their criterion because they were just expecting stimuli to occur. It was like the default. And the other way around, when there were less stimuli expected. Now, of course, she did simultaneously EEG, and now we can answer the question. Now we don't look at spontaneous modulations of anything, but we look at the difference between high and low criterion induced by a top-down expectation. And if she now compared EEG between these two conditions, what she found is that the decreased criterion went along with nothing in alpha, but a decreased pre-stimulus beta. So, of course, this is not the final proof that beta is not related to excitability, but it seems to be a different mechanism to be distinguished by different neural correlates. So I think it's based on this fair to speculate that while alpha changes the excitability, the overall excitability, and also has a noise suppression goal, it could be that beta is really closer to a decision threshold, and so that there are two kind of thresholds to be overcome that as I said, can also be reflected both in signal detection theory, but with different neurophysiological underpinnings. More work to be done, of course. So back to this picture, I was telling you a lot about neural correlates of entering and exiting the gray zones, neural correlates of the zone itself. Of course, all these findings were based on a certain definition of the gray zone. And now I would like to challenge the definition of the gray zone and introduce some behavioral findings that introduce maybe a more, even a more fine-grained distinction of what we call the gray zone. So this is actually one definition, as I said, so perceptual awareness, unaware, undetected, and then subthreshold. But one question arises is, how unaware are we really of these stimuli? So is there maybe some aspect of this information that we can get access to? And this reminds us, of course, of the phenomena of blindsight. And you know that blindsight patients with infarction in the occipital cortex and this hemifield deficit, this hemianopia, they can still make correct statements about stimuli in this field where they are blind and where they always say, I do not see anything. And it's Israel that performed experiments to see whether we might be able to find a similar phenomena actually in healthy volunteers, in stimuli that people do not report as have detected them. So what she did is the following. She did an experiment on detection as before, but she stimulated two fingers, one of the two. And then subjects were asked, did you feel it? Yes or no? And then even if you did not feel it or felt it, you were always asked which finger. And what she finds is the following. So here first, with increasing stimulus intensity, obviously the detected yes-no answers increases. And also, obviously, the correct localization increases. It starts at 50% because if it's a two alternative first choice task. If you guess, you are at 50%. Now, interesting is the figure on the right side. Now, if we distinguish those trials where people said, yes, I felt it, and then checked how often they were correctly localizing it, it always also increases with increasing intensity, soon reaching almost 100%. But here are the undetected trials. Those about 50% of the trials where subjects said, no, I did not feel it, but still clearly higher than 50% detection threshold and increasing with intensity, they correctly localized the stimuli. And Esra did another experiment where she did not perform two finger stimulations, but where she looked at two intervals. And the subjects then had, in a 2 AFC task, decide in which interval the stimulus was performed. And she had similar results. So to make a long story short, what we find here is that we can actually have access, at least partial access, to stimuli about which we say, no, we have not detected them. So what does it mean for our definition of the gray zone? So it means that now we have to distinguish between stimuli in this no awareness zone, which have correct localization, and stimuli which have no which also have no detection, but which have an incorrect localization. And of course, if we now start to distinguish between these graduations of not being aware of something, you can also look at neural correlates, and very early study, but what Esra seems to find, and it looks very interesting, is really that you can distinguish between stimuli that you perceive. Okay, that's clear. So this is the green curve. But if you compare miss, which were correctly localized, versus miss, which were not localized, you see in source localization in the superior parietal cortex, you see that there's a difference. So there, the correctly localized stimuli have a clearly higher signal than the not localized signal. And just to confirm previous results, both don't have an N150, so both have no indication of being consciously perceived. So, of course, more studies have to be done on this, but this is quite exciting. So we can actually differentiate neural correlates of actually graduations of the gray zone where we have at least partial access to it. So this means that our gray zone has now at least two different definitions, and yet more definitions are possible. For example, in recent studies, we have started to include metacognition. So when you make a decision about perceptual awareness, then you can ask the subjects, are you confident or unconfident? And the same about misses, are you confident or unconfident? And so all of a sudden, you have 4 different categories, and the gray zones could perhaps also move into this area of perceptual awareness when subjects say yes. Actually, Martin Grund has done a study on this, and interestingly, so not yet on neural correlates, but interestingly, he found a marker to distinguish confident versus unconfident stimuli, a quite surprising marker in a way, because in this study, he also recorded EEG, and we all know that after a stimulus, many stimuli, there is a transient slowing of the heartbeat. And we actually previously thought and also reported that this slowing of the heartbeat is more pronounced when subjects have a conscious perception versus not. But what it turned out now, it seems that the major determinant of the conscious perception is not of the heartbeat slowing, is not whether you perceive it or not, but whether you are confident or unconfident. So this contrasts the heart slowing of unconfident and confident stimuli. And if you look here at the confident misses, they have a similar heart slowing, or let's put better here, similar heart slowing like the confident hits, but it is the unconfident miss and the unconfident hit that have a more pronounced heart slowing. So even the body seems to react differently to a difference in confidence. And we are now looking obviously also at neural correlates, which I can tell you more hopefully in the near future. So there are different definitions of a gray zone, and maybe there are even more. And of course, with each of those definitions, we are now can investigate correlates of confidence, correct localization, with partial awareness, and so on. But this last, the metacognition and its relationship to the heart slowing, brings me to my final brief point in the last few minutes, basically emphasizing the point that the brain, of course, is not an isolated thing, but the brain actually does everything in close interaction with the body. This heart slowing is one of the indications, but there's more about this, as we have recently heard or found in several studies. So in our first study, it was Pavel Motika, a visiting student, who simply did also look at detection of near threshold stimuli, but he recorded simultaneously an ECG. And then post hoc, he checked whether stimuli that just happened to be applied during systole are more often or less often detected than stimuli that happened to be applied during diastole, late in the heartbeat. And lo and behold, he found that the hits are much more focused in the late phase in diastole, whereas the misses are much more focused early on in the systole. And so the phase of the heartbeat seems to determine the access to stimulus awareness. And later then, Esrael very systematically looked at this finding. She confirms the main finding. So here you see a contrast between systole and diastole. And you see that in diastole, you have significantly more you have significantly more hits than in systole. And actually, interestingly, in this case, it's not the criterion that changes, but in this case, it's the sensitivity that differs between the two conditions. And also, interestingly, this is a task where she also did this numb touch, as we call it, experiment. So basically, you localize the stimuli, even if they have not detected them. And actually, the localization ability did not differ between the two conditions, but rather it seems to be something that again regulates the access to detection, to awareness. And Esra also looked at the evoked EEG activity and found no changes in the early components. And 140, interestingly, also is unchanged between the two conditions. However, she found differences in the P300. I'm not sure whether I mentioned the discussion about the P300 before. So some people thought P300 is an index of conscious perception. Others, for example, Felix Blankenburg's group has recently provided very good evidence that it's actually not an index of conscious detection, because if you have a no report task, then the P300 difference between detected and undetected disappears. So in this case, we have a P300 difference. The question is, what could it mean if it's not awareness? It could actually mean, and that's our hypothesis or our explanation, that in systole, stimuli are suppressed because the body expects some pulse-related signal, noise in the body, and therefore suppresses this. And so basically in a predictive coding concept, stimuli are suppressed, and basically then this would mean that the P300 has something to do with the prediction of a stimulus, which is then which is reflecting this prediction, and which then differs between a stimulus that is predicted and that is not predicted. It's a interpretation to be confirmed in further studies. Interestingly, Esra had another interesting observation relating the heart to evoked activity. It's been known for some time that there is a representation of the heartbeat, of each heartbeat, in the brain, the so-called heart-evoked potential. It has been related in previous studies to the relationship, the balance between interoception and exteroception. And consistent with this hypothesis, Esra finds that during misses, the heart-evoked potential was much smaller. The heart-evoked potential preceding the stimuli was much larger, more pronounced than before hits. In other words, before a miss, we were more in this balance on the interoceptive balance, whereas during hits, we were more on the exteroceptive balance. So the heartbeat evoked potential is stronger preceding misses than hits. And she, of course, again looked at parameters of signal detection theory. And in this case, the HEP, the heart evoked potential, was related to, was not related to sensitivity. Detection changed, as I said, but it was strongly again related to the criterion. And if you look at the evoked potentials, then you find that already the P50 so already early components of the evoked potential changes, which seems to indicate could be an indicator of higher or lower, in the case of of low heart evoked potential, higher excitability. And we tested whether it's related to alpha. Actually, it's not related to alpha. So it seems that this balance between interoception and exteroception is an additional parameter which can help us access a signal or suppress a signal. Also to be investigated in the future, but I think it's quite exciting that such a heart-related signal relates very early on to sensory processing. Now, finally, Martin thought the heartbeat is very closely related to respiration. So is also the respiratory cycle related to our perception? And what he found is actually that respiration is related to stimulus performance. And actually, it seems that all stimuli are dominantly located in a certain phase of the heartbeat, of the respiration and the early expiration. And you can also see that the trials and the distribution of trials closely matches the effect of the respiration on the heartbeat. So during each respiratory cycle, the heart frequency changes. This is IBI, interbeat interval. So the heartbeat becomes slow and then faster again. And this is precisely matched in the localization of the trial. So it seems that the respiration is tuned to have the stimuli at a certain phase in respiration. And it was Daniel Kluger in a recent study where he showed that actually respiration aligns this perception with the maximum of neural excitability, actually indexed by alpha. So respiration really modulates alpha throughout the respiratory cycle and aligns it such that it's tuned for optimized task performance. So bringing the neural correlate, the alpha, and respiration and heartbeat together. So I'm at one major message is perception of near threshold stimuli is strongly modulated by bodily signal. And now I'm coming to the end of my talk. So I have talked about the gray zone of perception. I have shown you that there are different definitions of this gray zone. And interestingly, there is a phenomenon of what I call numb touch, that I have access, like blindsight, to stimuli that I do not consciously perceive. And that, of course, this relates to different definitions of this gray zone. Then I discussed the neural correlates of different stimuli depending on how close they are to our conscious awareness, specifically also undetected stimuli that lead to a deactivation and actually modulate excitability. I was discussing our ways how the brain might regulate stimulus perception and alpha, which I think there's now convincing evidence that it regulates cortical excitability, subliminal stimulation modulates it, and I think I would argue a well-founded hypothesis that beta could be related to a decision threshold. And finally, I showed you that the body is much more important than we previously thought. It also has strong influences of these simple somatosensory perceptions interacting with the neural correlates. So next time you looking for your classes, take a deep breath, think of me, and then you will find your classes. Let me finally thank all the people who have contributed throughout the years for this work. Vadim Nikolin, Esra Al, Martin Grund, Till Nierhaus, Karina Forster, Birol Taschkin, Pavel Motika, Ruth Schubert, Fivos Iliopoulos, Christopher Gundlach, Gabriel Curio, Tilman Stefani, Isabel Barreiter, Felix Blankenburg, Norman Forschak. They all have dived at some point into this gray zones of perception. I thank them, and I thank you for your attention. Thank you.
Thank you very much, Arno, for this very interesting talk. we already have questions actually. So we can, Jian, are you here?
Yeah, I'm here. Yeah.
Okay, Jian has a question. So Jian, please just go ahead.
Yeah, it's a very interesting studies. So one question I have is, would you like to make a comment on the role of the thalamus? I would speculate that, you know, when the signal comes from peripheral, then the thalamus actually maybe the key node to decide will the signal be go to, you know, go up to the sensory motor cortex to be detected or not. So yeah, would you like to make some comment on the role of the thalamus? Thank you. Yeah.
A very, very good point. Very good point. I mean, yes, the thalamus, as you all know, is kind of the entrance gate to the brain. And I mean, yes, I, of course, I simplified the story a bit. Obviously, these rhythms, for example, the alpha rhythm, is a rhythm between the thalamus and the cortex. And so, alone, if you think about that, it means that rhythmic activity between subcortical and cortical structures determine the axis. And yes, I would believe, I cannot prove it, but I would believe that some gating occurs already at the thalamus, but it's just difficult to examine, especially in humans. But you're completely right. I'm pretty sure that it already starts at the thalamus, if not even lower.
Thank you. Yeah.
Alex? Alex, can you hear me?
Yep, I'm here. Can you hear me?
Yes.
Okay, great. I have a couple, so feel free to cut me off if need be, but this talk was really fascinating. Thank you so much. So I was really interested by the similarities that you found across modalities that you described more towards the beginning, and I'm curious if you or colleagues also tried validating with auditory stimuli.
No, I'm afraid. I mean, we are looking every once in a while at the visual domain. Auditory, I have not done much, to be honest. I like to make music, but the auditory domain has not. I just have to say no. I'm sorry.
Okay, no problem. Maybe in the future. And
yes.
I'm wondering, I'm sorry if I missed any comment you made about this, but I'm wondering if you think your findings generalize from, you know, the emphasis was on stimulus detection, but I'm curious about how you think it might generalize to decisions about features or categories like a same or different task.
Yes. I cannot prove it. I can just try to extend our interpretation. And my strong belief is that it simply generalizes to also these other tasks. But of course, it will be modulated. No? So, I mean, the simple detection, yes, no, of a stimulus, of course, is something where alpha could play an easy role if it just generally regulates excitability. If you go into domains of differentiating, then it must be a bit more complicated. But there are indications in the visual system, for example, that you have. So, for example, if you differentiate tasks between different locations, that you might have a localized alpha increase in a certain location, hiding that kind of, and it's more, the excitability is higher in a different location. So a differential modulation of alpha in different locations could actually shape also or help the discrimination between two different stimuli. But we have not looked at this carefully in our domain. It's just an extrapolation of our findings, considering also some other findings.
Yeah, do you feel that could be some next steps, or do you think that there's more to be done before going down that path?
I think this is the next step. I mean, there are some findings I have not talked about, which keep me puzzled,
Mhm.
but could go in that direction. So, for example, we did some time ago, Norman did a study, Norman Forschak, where he looked at the question whether alpha modulates or is a mediator of the effect of attention. And the answer is no. I mean, attention modulates alpha, as you know, but alpha was not a simple mediator. However, what he found is that the relationship between alpha in bins and detection was different whether whether there was a focus of attention or not. In other words, so there seem to be ways how the effectiveness of alpha can be regulated. I don't understand it yet, to be honest. That's why I only talk about it now in the discussion. But this seems to go in a direction that, so because here there was differentiation between the stimulus on the left or right hand, but this seems to go in the direction of differentiating, discrimination of different input, and then probably the effects of alpha, maybe also beta, are then more complex.
Yeah.
Thank you very much. We have a lot of questions, so I have to cut you, Alex.
No
Please
problem.
I mean, if you have more questions, I will be very happy to be contacted personally. My email is easily available.
Sounds great. Thank you.
I think the next was Isaiah, as far as I remember.
Thank you so much. A quick question. You discussed earlier in the talk about the role of the insula activation in accurate detection, and then later about heart rate and respiratory rate. In terms of the direction of influence, do you think that it's insula activation, which is increasing the likelihood of accurate detection and also increasing heart rate and respiratory rate, or is the direction different?
Good question. Good question. I mean, it plays a role for both. It plays a role for both. And so my intuition is, yes, when it plays a role for having stimuli getting access to the conscious experience, and it seems also to code the insecurity or the lack of confidence, and that would then be the top-down mechanism for example, modulating heart rate. But we have not directly proven this. I cannot directly address this. What I can say is that I haven't talked about this so specifically. If we look at the heart-evoked potential, and if we look at the location where it differs between high and low heart-evoked potential modulating sensory detection, then we are right at the insula in dipole localization.
Thank you very much. Doug?
Hi, great talk. I have a question about pharmacological interventions, and have you investigated any pharmacology and its effects on these detection thresholds, and in particular around caffeine? Because if I remember correctly, caffeine is supposed to do something to alpha, so maybe it has a secondary effect on these detection thresholds.
We did some time ago. We did a study with Zaleplon, modulating GABAergic neurotransmission. The problem was that it's, you give a drug systemically in human volunteers, and then you have effects everywhere.
Yeah.
And you don't know. So, in the animal, you might, apply it locally, and then you know where. And it was very inconsistent, I have to admit. Caffeine is interesting. We have not looked at that. Should be easy. I could be a good subject for that. But no, we have not looked at it pharmacologically.
Thanks.
We have then Nushin, I believe. We will have 2 more questions. Yeah, please.
Hi, Arno.
Hi,
Thank
Nushin.
you. That was a really good talk. I have a question. Do you think that people who have hypersensitivity to certain stimuli, like touch, sound, or smell, like you can see in autism, would have different alpha or beta signature? And do you expect that would be modality specific?
I can only speculate also, but I'm pretty sure that these regulatory circuits should be affected in people who have a hypersensitivity. So I think it's a very reasonable way to proceed to invest those people with autism, for example, for hypersensitivity or for lack or for disturbance of the regulatory mechanisms. Yes.
Mm-hmm. Thank you.
Maria?
Yeah, thank you. Thank you for the great talk. Arno, I just have quite a naive question, probably in the continuation of this question from what is the direction for the interoception and exteroception. So when we're saying that during different periods in the heartbeat, systole and diastole, for example, we have different sensitivity for stimuli. So do you mean in this case that it's actually the modulator is really the body, and can this cycle can it be reversed in different pathological conditions, or can it be reversed, for example, in different body specific psychotherapeutic, for example, practices? So can we move it? Probably it's, it's not articulating it well.
Thank you. I understand what you mean, Maria. So the phase of the heartbeat, this effect is most likely mediated by baroreceptor activation. And so it's not, I don't think it's related, this effect is related to interoception or exteroception, but it's most likely mediated to baroreceptor activation. And I think this effect, that baroreceptor activation leads to a modulation of sensory input, could also be very relevant for certain diseases. So, I personally am pursuing the hypothesis that for the development of hypertension, this could be a quite important factor, because baroreceptor not only mediate modulation of sensory perception, but also well-being, emotional perception, perception of stress, and so on. So there it could have a real implication. And speaking of disorders, I would also assume that in heart disease, this effect should be significantly modulated. Now, regarding interoception, exteroception, that is related to the heart evoked potential. And there, yes, the simplified interpretation is that with a higher heart evoked potential, we have higher interoception, and therefore we are less susceptible to stimuli from outside. Again, it could have implications for diseases. So we just finished a study, Denis Kumral. We finished a study on patients with atrial fibrillation, and they have which is a heart disease, and they have a dramatically reduced heart evoked potential. So of course the question is what does it mean for the disease or for the disease process? I actually think it's also related to disturbance of heart-brain interaction. But it tells us that these simple sensory signals really could have profound implications as proxies for disease processes.
You're right. Thank you.
Thank you very much, Arno. We have a lot of questions. I have a lot of questions, but for the sake of time, we have to wrap up most probably. And for the other people, please just, as Arno said, just contact him directly. Thank you very much for being here. It was very interesting talk, and see the other people in the next week in the other BrainMap. Thank you.
Thank you very much. It was a pleasure for me.